Let's do it.
Okay, so actually, is there water around here?
Huh? There's water fountain, but I don't know
I
about
hope this.
bottled
I'm gonna
water.
lose my voice at some point, then I'll take a break and get some water.
Okay.
Do it that way. Okay, so I'm Alex Rubinsteyn, and I have a research lab, but it's actually kind of a weird structure. So me and two other researchers at UNC formed a joint lab. This is not really recognized by UNC. Like, we kind of get like a lot of hassle for doing this. So we have to like technically apply for funding separately, do everything separately institutionally, but then we just pool all of our money, hire scientists together, and just work as a collaborative lab. It is the Personalized Immunotherapy Research Lab. There's a new picture with everyone together, so I should get that for these slides. And so this is like one of my grad students is just AI generating the future that Pearl wants. And so like very egotistically, like we like actually want to cure cancer, which I know is in some ways is pretty taboo to talk about because so many people work on it, so it seems like intractable. But we want to work on stuff that feels like plausibly this is the path to cure cancer. There's a lot of stuff you can do in cancer research which is not that. So if you work on something with a really slow timeline, so you work on developing a small molecule drug that has like a tiny patient population, that's not like a path to a general purpose cure for cancer. If you work on like extremely fine-grained characterization of a specific cancer type's genetics, important, but not a general path to curing cancer. So we like want to do something that feels like it ends up here. Even if we don't end up making the nanobot armies that cure all the cancers, we want to do something that kind of feels like when someone gets cancer in 100 years, you do something, and little nanobots go and they kill all the cancer, and then you're fine. And so the nanobots that are readily available to us for engineering and manipulation are elements of the immune system. So we don't have to actually make little robots with gizmos and tentacles and lasers and stuff. We already have things that sort of do this, which are all our different immune cells. And they're crazy. Like, immune cells are extremely destructive. When they're sort of pointed at the right target, they do things that seem sort of miraculous. The turnover rate of neutrophils, you generate like, I don't know how many neutrophils a day you generate, like 1 trillion or something. And they're little suicide drones that home to any source of infection, and they physically blow up and they send nets of DNA to cover all the bacteria and viruses. And T cells have this sort of like also tremendous reproductive capacity, amazing capacity for tissue destruction. If it's a cancer, they can, if they're really pointed at, wipe it out very rapidly. So it feels like if the future is, if someone gets a cancer, we want a nanobot army to kill it, we kind of have the right materials. We just need to figure out how to point it. So I'll do a little bit of background of using the immune system to kill cancer. So oncology kind of starts in the neighborhood of these kinds of therapies very early on. So if you go way back in the literature, you start seeing observations of a relationship between the immune system and cancer in the 1800s. So Chekhov actually wrote about a patient he had who had a cancer that then developed a fever. This is like 1870s in Russia. And then after the fever, the cancer seemed to start shrinking. And so it's kind of an anecdotal observation. Oh, like something about the infection or the response to it seems to have cured this cancer, or it seems to be at least affecting the cancer. And so these anecdotal observations start getting kind of like honed towards a specific therapeutic hypothesis by William Coley, who was in Memorial Hospital, which eventually becomes Memorial Sloan Kettering. And so he had first a live culture of bacteria, and then eventually a sort of purified set of toxins from the bacteria that he would inject into sarcomas. And it actually sometimes worked. So he had like a 10% cure rate for sarcoma, which is a very tough, broad category of cancer that's all sorts of weird things. But even now, like a lot of sarcomas are pretty intractable. So in the like early 1900s, so like 1907 or something, he publishes a case series of patients he treated with what he called Coley's toxins, and he had a non-trivial cure rate, and that was really interesting. However, it was hard to reproduce because he had his own bacterial culture. Other people started using other bacteria. It was finicky, it it wasn't really precise enough that this could be scaled up. And then people started doubting whether it was real in the first place. You know, if it worked in his hands, not in other people's hands, maybe the whole thing was fake. And then we start getting precise therapies, so sort of therapies that are not so finicky and biological, but they have a measurable dose-response relationship, which are radiation and chemotherapy. You zap the tumor and it shrinks, and maybe it comes back, but you can see the relationship between your therapy and what happens. And so that creates what I kind of think of as the dark ages, where oncology becomes you poison the patient and radiate the tumor. That went on for really quite a long time with not a ton of progress in curative therapies. Like there's specific cancers for which you get curative chemotherapies, but there's this huge swath in which it was all sort of small incremental bits of progression-free survival benefit decade after decade. So like metastatic melanoma, despite many different trials and chemotherapies for metastatic melanoma, remained largely a death sentence until cancer immunotherapy. So cancer immunotherapy in its modern incarnation is mostly these drugs that are called checkpoint blockades. They're not Coley's toxins. They're not the kind of therapy I work on. They're drugs that disinhibit T cells to go and kill a cancer. So this is a melanoma trial, and this blue curve, both curves are essentially survival over time. So when the curve goes down, someone dies. And so this blue curve is the previous standard of care for melanoma, which is this chemotherapy called dacarbazine. And then the yellow curve is an immunotherapy. So it targets PD-1, which is whatever, doesn't matter. Makes T cells more aggressive. And so kind of a qualitative difference between these styles of therapy that really grabbed everyone's attention was that the immunotherapy curve is an asymptote. So it's not working for the patients up here, but for the patients that it worked for, it continues working for. So it gives them durable benefit. Sometimes curative, sometimes there's like a détente between the cancer and the immune system, but the patients survive for a really long time. So a lot of people took notice of that. Now we've had like Nobel Prizes for this, and so it's really like a big deal. And I went to the American Association for Cancer Research meeting like way after I got into this field. So I started working on cancer in 2014, and I went to the ACR meeting in like 2014 is when these trials were dropping, standard of care was flipping, and I finally went to AACR in like 2019, and they had like a chemotherapy section, and then they had like 10 rooms that are all like it was a total reversal of how oncology research had worked previously. And I think a lot of the past decade has been like fleshing out like where can this be applied, because it's not everywhere. There's cancers where this doesn't work at all. There's cancers where it sort of works, but you get the asymptote is at like 30% of patients benefit. So it's not a general purpose solution, like just making T cells angrier is not a general purpose cure. But for some cancers it actually is. And in other cases it boosts the long-term survival from like 5 to 30%, 5 to 40%. So strong hint that the immune system is potentially like the right place to be looking for new therapies also. Okay, so the effector population of interest here, there's a lot of different immune cells. So there's T cells and B cells and natural killer cells, which are a great name for a thing that sometimes does what it sounds like. And then there's all these other like neutrophils and eosinophils or whatever. So the T cell seems to have the largest role in cancer. There are other immune cells that have roles. Some immune cells have sort of negative roles. They're like pro-cancer, cancer can recruit them. But the T cell seems to be like the main kind of like soldier that's on your side in a fight against cancer. And its role is essentially like to be, it's like kind of like it's like living in a police state, like a very xenophobic police state maybe, in that T cells go around and they surveil all of the tissue of your body. They circulate, they leave your vasculature, and they home to different areas following different cytokine gradients. And they're very hands-on, constantly sensing the surface of every cell. And they're essentially looking for, they're like implementing this outlier detection algorithm. they're looking for stuff that looks out of place. And if a cell looks sufficiently out of place, meaning it's making stuff that's out of place, it gets killed. So the canonical out of place thing to be making are viral proteins. And this is kind of where T cells got characterized. Oh, they look like they're sort of like an antiviral immune system. And they can adaptively figure out different sorts of useful patterns to go after. So you get a viral infection. Initially, your T cells don't know about it. They don't know to kill cells that are making that pattern of proteins. But once the virus has done a little bit of damage, then T cells get licensed to expand and go after it. And they leave behind a memory population that the next time you get the virus, they expand faster. And so you get T cell immunity against the virus. And so they can play a similar role against tumors. The tumors have to be sufficiently weird. So the weirder the cancer, the more likely that they sort of trip the like outlier detection threshold for T cells. So melanoma, which is getting zapped by a huge nuclear reactor in the sky all day long, often looks weird. And so that's like, similarly, lung cancers that are exposed to smoking, that have been carcinogized extensively, they often look weird. So these are the kinds of cancers where T cells will naturally take notice, or they'll take notice if you sort of nudge them a little bit. There are other cancers that have simpler mutational structure where T cells can't figure out that they're different. So the cell looks the same. So it doesn't have a pattern to lock onto. Okay, so the mechanism for that kind of pattern recognition, and I'm trying to not get too far into the immunobiology, but on the T cell side, every T cell is a unique receptor. On the cellular side, the cells have to essentially show this kind of like binning histogram of their protein contents on little presentation platforms called MHCs. And if they don't follow the protocol, if they don't show enough of like what are you making on an MHC, then NK cells, which are kind of partner to the T cells, come by and kill the cell. So you have to follow the protocol. You have to, whatever your protein contents are, you have to dice up those proteins and put parts of them on the surface for T cells to inspect and see if they're weird. And if you don't have enough of that on display, then NK cells come in from the side and kill you. So that's why I think of it as like a police state. It's a very like papers please, and if you don't have the papers, you're in trouble situation. And so this recognition is, the adaptivity really comes in terms of the part of the protein that's getting shown to the T cell. And the T cell receptor is specific to just, well, it can be like up to like 1 million patterns, but out of a sequence space that's much larger than 1 million. So there's a lot of specificity in that recognition. And the population of T cells gets trained by looking at the sort of normal proteins your cells make, and the T cells that recognize normal stuff will die or enter sort of inactive states. And the ones that don't see, like their TCR doesn't find anything on normal cells, they're sort of allowed to go and kill their targets. So that's the outlier detection algorithm as the body implements it. Okay, so this is the hook into what I'm trying to build up, which is this extremely personalized medicine called neoantigen vaccination. And so the idea here is if someone has a cancer that has grown, it has evaded that T cell surveillance. And if it's not of a cancer type that we think will easily get cleared by checkpoint blockade, where just this sort of aggressiveness nudge alone is not going to be enough, maybe the problem is that its surface area of possible targets is a little bit too subtle. Like you need a lot of weirdness for T cells to figure it out. So we can give the T cells a big hint through therapeutic vaccination. So it's not like prophylactic, and it's not trying to get antibodies. You're trying to get T cells to raise up a population that recognizes some specific targets from the cancer, and then go around and kill everything that presents that. So we're gonna exercise some like high-level judgment that we think the T cell compartment in its distributed algorithm might have missed. You know, it didn't know to look after things that have these 3 targets, but it should have, so we're going to tell it to go after that. So that's kind of the high-level idea. And as a therapy, this is logistically pretty crazy, because what you have to do is you have to synthesize the therapeutic on a one-off basis for every single cancer. And so that's not really how medicine currently works, but there's a lot of existence proofs that you can do this. It's just a huge pain in the ass. Like I worked on 3 trials that did this. Lots of trials in the past 10 years have done this. It's very expensive and annoying, but you can do this. If it worked, then it'd be worth doing. Currently it does not work. So I can talk a little more about that. But this is kind of what I mean in terms of like, my lab is interested in stuff that feels like it can be on the path to curing cancer, because these kinds of therapies, if you could get them to really work, could be the thing that in 100 years is just really routine. Oh, there's a malignant growth, okay, scan it, make the vaccine, next day get an injection, it goes away. And then cancer stops being a big deal, hopefully. Okay, so in the universe of cancer immunotherapies, I work on the currently useless one. It doesn't really have any success stories. There are lots of claimed success stories, they're mostly bullshit. Checkpoint blockade has like big success stories, hugely transformative in tons of cancers, like massive benefits in overall survival. CAR T is like really impressive engineering, and for a few cancer types has made a big difference. It's a lot more niche, so it's a kind of therapy where you, instead of trying to educate T cells about what targets to go after, you just pick a very specific target and you just weld an antibody fragment essentially into the T cell, and you tell it, this is your new receptor. You're gonna kill stuff that matches this pattern. And because all that engineering has to happen beforehand, you get to pick just a couple shared targets. So the kinds of cancers you can kill that way have to have some shared vulnerability, and what that is shaken out to is you essentially have a bunch of different surface markers of B cell lineages. So for different B cell cancers, you can like nuke the whole compartment. You can kill all the plasma cells, you can kill everything that's at all like vaguely resembles a B cell. And then patients who get these therapies essentially have a living drug that goes around where these engineered T cells kill the B cells. It's like a nasty civil war in your immune system. But within that population of B cells, there were cancer cells, which are now gone. So it's an effective therapy. It is limited in what it can do, because so far it hasn't worked anything other than these sort of like hematological disposable populations. And then vaccines have this like nice theoretical story for what they could do, but there's no success stories, which sucks and I want to fix. There was a whole bubble of cancer vaccine work from the 80s to the early 2000s. And there's a couple reviews showing the outcome of that. There's a bunch of trials with different vaccine platforms and different targets. And this is a column showing any measurable response in patients' tumors to these vaccines. And it's like pages and pages of 0 with like occasional single-digit non-zero. So this is kind of the like state of cancer vaccines in like 2011, 2012. They're kind of an exciting modality that felt debunked. And then there's a big comeback and people started funding cancer vaccines again. And so the reason that happened is essentially high throughput sequencing. And so instead of having a small number of shared targets you go after, you can start discovering mutational targets in a patient-specific way. And there's this idea that neoantigens, so like a immune target that's new per patient, per tumor, are going to be better targets because they're mutant and they're not self, and T cells will treat them as sufficiently foreign. And so these tumor neoantigens will be great targets, and then we'll make vaccines that target them. And that's kind of the like pitch that I was given when I started working on this stuff. And as a naive computer scientist, I was like, great, we cured cancer, let's do it. I'll write the Python code, we'll start the trial, good to go. So I did write a pipeline at Mount Sinai. It's all open source, you can use it if you want. It's being used by Break Bio and a few small startups that I tell that it's not gonna work and they kind of ignore me because it doesn't help their pitch deck. And so yeah, you can use this pipeline, and we ran 3 trials off of it. I think a 4th trial after I left got started. They had kind of a similar design. They all did tumor normal DNA sequencing and tumor RNA sequencing, run all that sequencing data through the pipeline, the OpenVax pipeline, pick stretches of the mutant proteins with the mutation kind of centered. So often the changes were just single amino acid changes, so you gotta stick the change near the center. Sometimes you slide the window around because what you can manufacture on a quick timeline is tricky. Like, we're trying to manufacture a never before made drug in like 4 to 8 weeks. And then we're gonna inject it in the patient after. So there's a lot of manufacturing heuristics around what we can and can't make. And then it gets mixed with this adjuvant poly-ICLC, which everyone assured me is extremely good. I didn't know anything, so I said sure. And the FDA loves it because it's safe, and so they'll let you do it. I'll just say that this trial just totally bombed, and this trial also pretty much bombed. And these numbers are outdated, so they accrued more people. So this one actually ended up with 12, this one got 10. The first and the third trial, like, everyone progressed, it wasn't really useful. The middle trial might have worked, but the 2 PIs of the trial got into a fight. And so now they won't like share data or like do any of the stuff you need to publish it. So I'm like very interested to find out if the small tweaks in that trial actually made a difference, because there were some small differences. But yeah, I don't know. Can't find out because I think they're currently suing each other. Possibly because maybe it worked, so it's worth money, I don't know. But the first and the third trial definitely failed, and a lot of other trials that have a very similar flavor failed. Like, they just didn't do anything useful for the patients. So do these vaccines work? I think I already told you they don't. There's a lot of papers on these trials, not just ours, like tons of other ones. So this trial is the one that I think we copycatted. Had same adjuvant, same peptide length, and they had something that was like very suggestive of it worked. But as you kind of dug into it, you realize that they got responses in really like these 2 patients for whom, like, there's patients for whom the standard of care therapy was probably fine and they weren't going to recur anyways. And they had 2 stage 4 patients that were going to recur, and they give them checkpoint blockade, which works for melanoma, and then they responded, and then they concluded, and thus their vaccine must have worked, which is like, it's pretty fishy. There's a lot of papers like this that have sort of a similar, like, they pile on therapies where you know you're going to work, and they add the vaccine, and they assume the vaccine was the active part. There's a kind of a tell, I think, to like the vaccine probably didn't do anything, which was a subtle color coding to their T cell responses that took a while to figure out, which is the ones that do the killing, these CD8 positive T cells. The reason that these boxes are yellow, if you go through the supplement, turns out to be that they weren't detectable responses from the blood, the way that the helper T cells over here were. They needed to be expanded for 3 weeks, which means that the population in the blood was probably like 10, 20, 30 cells or something, you know, some tiny number that was definitely not going to be sufficient to go and home to a tumor and kill it. So that was like, I remember starting to kind of doubt some of this stuff, looking at this paper and then looking at our immune monitoring protocol and thinking, we also do a really long expansion to see CD8 T cell responses. And these are the ones that are supposed to be killing the tumors, so that's like, that doesn't feel like it's adding up. They did a trial, it's not exactly the same group, but there's an overlapping group of researchers that did a trial in glioblastoma. They essentially showed that if you have a T cell based therapy and you give the patients steroids, then it definitely doesn't work, which, like, in hindsight maybe is obvious, but I guess it's good that somebody did a trial. Also, glioblastoma is very deadly. And in our trial, half the patients are alive, and I really wish I knew if the vaccine did anything. Whatever. There's an mRNA vaccine that had sort of a similar story where they had CD8 responses after in vitro stimulation, also not ex vivo detectable, also kind of a hint that maybe even with mRNA, the T cell responses are not enough. And then there's a company called Neon that did a fairly large phase 1 trial of a vaccine design similar to the one that I worked on at Mount Sinai. And they took on, I think that this is not accurate, I think they took on like $300 million of investment and then got sold at a steep discount to BioNTech. Possibly also hinting at how well that particular kind of neoantigen vaccine is working.
Or not well.
Yeah. Okay, so I want to diagnose some problems in this field because I think that the general scheme of this kind of therapy is a good one. Is a good idea for like a long-term goal as a field, but it got actualized all in the mid-2010s with very specific decisions that were convenient for the technology available then, that I think left behind some huge holes that keep the therapy from like fitting together into working parts. So one is that if you're doing tumor sequencing in like 2015, 2016, you have a Illumina HiSeq 2500, and you have kind of limited memory and storage capacity, and you don't really want to do a whole genome sequencing, and then this idea of exome sequencing feels like a huge bargain. So everyone starts doing exome sequencing. And definitely you're going to do Illumina because the long read stuff was extremely embryonic then. So nanopore sequencing was really, really high error rate. PacBio was pretty high error rate. I don't know if they had consensus reads yet. So stuff where you do long read felt like extremely exotic. It would cost you like $100,000 per patient to do like a half decent job. Whereas with Illumina short read exome sequencing, you could sequence patient, tumor, and normal and RNA for like $3,000. And so that's the standard solution. And so everybody started doing the exact same thing. No matter what informatics they ran, they would do more or less the same. You do some SureSelect exome capture kit fed to Illumina HiSeq 2500 and poly-A capture of RNA, and your read length is like 100 or 120. Like the sort of data source is all identical, and then they run informatics on it, hoping to then recover more information. So like Neon had like all the variant callers you could find when they built this pipeline, but the results are pretty much identical because the data source is the same, with like the same kind of biases in what you're missing. So this goes beyond even vaccines. So there's a cell therapy lab, Steve Rosenberg's lab, that does this kind of unbiased screens of neoantigen responses, but it's unbiased after variant calling from exome data. So as long as the exome sequencing were a good representation of the mutations in a cancer, this could be considered unbiased, but it sort of isn't. There's another sort of T-cell response identification thing, also exome, whatever. Everyone does the same thing. I did the same thing, everyone does the same thing. So the problem is that what we actually want are targets that are pretty dissimilar from the self proteome, and exome sequencing is really biased towards single nucleotide variants. It has, first of all, the read lengths are short, so you're gonna miss larger indels almost by default. So if there's like a 3,000 base insertion, which now they're doing a lot of long read sequencing, is not that unusual to see, the tiling over that with short reads and assembling that novel thing and figuring out where the breakpoints are, that's like a project you could give to someone for like a year per mutation. It's not feasible to just figure it out quickly. So what you end up with is stuff that fits inside the read length. But you also need sort of like sequences on the side to align well, so you end up with stuff that fits well inside the read length. Like maybe your max for insertions is like 10, 15 bases, and after that, things don't really align that well. And then the exome capture itself has a really big bias away from large mutations because the baits for the bits of DNA that you're going to pull down that match the canonical exome, they don't work well if there's a bunch of novel sequences there. So exome sequencing ends up really biasing you towards stuff that's like pretty much the reference exome modulo a few flipped bases. So you end up with single amino acid substitutions as your predominant sort of mutation. And those are really similar to the self proteins. They're the self protein with like your valine is now an isoleucine. Okay, great. Like the immune system probably can't see that change. Whereas the thing that you ignored, the 3,000 base insertion that turns off the lights, that probably created a cryptic exon and completely changed the reading frame, you just can't see it all. So there's a couple studies that look at this that try to compare Illumina, nanopore, PacBio sequencing, and they all kind of find the same thing, which is that Illumina, just sort of the coarse grained characterization is that Illumina has for a really long time been high accuracy and affordable. And then there's all these other mutations that you can find with PacBio and nanopore, but they're trickier. The ONT data has always been sort of noisy, PacBio has been really expensive. So this is not true anymore. Like as of this year, like all these other mutations, these long read specific mutations, you can now actually feasibly get pretty easily, like cheaply without a lot of effort. But for a long time that was hard to get, so people didn't use it. They sort of knew that you're missing stuff with short-read sequencing, but then they say, well, but like the informatics of nanopore are tough, and PacBio will cost like $50,000, so I'm not going to do it. So with the PacBio Revio, PacBio got cheap. With the nanopore R10 flow cell and their duplex reads, where they do 2 passes over read, they have high accuracy. So all these like mutations that people were ignoring are now sort of in reach, but no one's really using them yet. And so that creates a lot of opportunities for like maybe the stuff we train the immune system to go after should be mutant proteins derived from all these other mutations. So one thing that I noticed we started doing some of the sequencing is that a lot of what gets missed are indels, so insertions and deletions into the genome, and they just span a pretty wide distribution of lengths. And the short read distribution of lengths like falls off almost immediately. So you can see indels that are like tens, hundreds, thousands of bases long, but you just don't see them with Illumina sequencing. You see them with other stuff. Okay, so whatever. Get too technical, did a mouse study. I'm going to show some just example, little screenshotty things of what these mutations look like. So this is a really big inversion. So it's like a part of the genome that just got flipped upside down, which creates like, who knows what that does. What we see is that that creates expression of something that has nothing to align to. So in the RNA, you can see that the inversion extends this transcript into this sort of like inverted sequence, but there's no tools that tell you what that is or what the predicted coding sequence of that's going to be. It's just some like intergenic junk that's getting expressed now. It's just a cryptic exon. So one thing my lab is working on is like, what is the tooling let's use this data to predict the mutant proteome when the mutations are not of the sort that people are used to annotating. It's not this single base substitution changed this amino acid in a coding sequence. It's like a mega base inversion created a cryptic exon somewhere where a gene used to not exist. That's much harder to figure out, and currently we don't have the tooling for that. But once you have that tooling, you can start actually seeing what's all the mutant stuff in the tumor that you're ignoring before. So another example. So this is a 470 base insertion that you could only really see if you look in the alignments. You have to look in the clipped reads. Often you see this stuff in the clipping. So a liner has this really tough job of taking all these sequences and trying to smush them onto a reference genome. And especially with RNA, there's a lot of uncertainty where maybe something can be spliced away. But so what the aligners often do when they're faced with large mutations is they just throw their hands up and they will just like skip exons and clip the ends of reads until they can say, this pretty much matches. And so when you're looking around the alignments, you have to start really second guessing the aligner and looking at all the soft clip regions. And what you find is that, so here is an inserted sequence in the DNA that is also in the soft clipped areas of the RNA. So it's getting expressed, it's just the aligner doesn't know about this mutation, doesn't know where to stick it. It doesn't know to sort of treat it as an insertion in the RNA reads. So this is kind of like a limit of informatic tooling. So you can do the sequencing, there's not yet good tools that'll make sense of the sequencing for you. Okay, another problem. So this is a study, I think this is Genentech did this study. They looked at 2 cell lines and they ran these kind of like neoantigen prediction pipelines with lots of layers of filtering in like 2014. And they did something smart, which is they looked on the physical surface of the tumor cells, and they wanted to validate the predictions, the things that are supposed to be presented to the T cells, are they physically actually there? And you could do this using mass spectrometry. So you can pull the MHCs off the cell and then change the pH to get them to sort of release the peptides. And then you put that in a column and feed that into a tandem mass spectrometer, and there's a lot of fancy physics that happens, and you can reconstruct the sequence of the peptides that the MHCs are presenting. And if you do that, you almost always get a funnel plot that's just like this, where like you start with lots of possibilities, and then you usually get to like 0, like nothing really validates, because it's really specific, and the predictions sort of suck. And so when this paper came out, it was just broadly doubted because the sensitivity of mass spec was thought to be low. And one trial tried to do this, and they also had trouble finding anything to vaccinate with. And so everyone just kind of like hand waved, oh, mass spec is low sensitivity, let's stop doing it. But like, we've been doing this with targeted mass spec, which has a much higher sensitivity, and it still looks just like this. Like most of the stuff you predict isn't there. So it's a deficit. On the one hand, if you had abundant tumor tissue and a really fast turnaround mass spec core, you could do this as part of any process to make a personalized immunotherapy. On the other hand, if you had machine learning, which didn't suck, you could obviate the need for this. So you could make an in silico simulation of the antigen processing and figure out what are going to be the 7 things on this cell line, and hopefully enrich this set of source mutations and get more than 0 on this cell line. But we don't really have those machine learning tools right now. So you either need to have like really fast, high throughput mass spectrometry, or you need better machine learning. Does not currently exist. So that's a big limit. And then the other thing is that, so I was showing you those CD4 versus CD8 T cell plots where the CD8s were like infinitesimal. There are vaccines where you get really strong CD8 T cell responses. So this is from a trial of a yellow fever virus vaccine. It's a live virus. And the detectable, just straight out of the blood, circulating population that recognized a particular peptide from a single protein of yellow fever virus, in this patient goes up to like 6% of just all circulating T cells, maybe all circulating CD8s, but you know, a really large number of cells. So a good vaccine looks like this. If they had done this and these were all just 0, and then they had to like fall back on like a 3-week expansion protocol to see any of them, they would have called this trial a failure. And so you want a vaccine platform that's like this, and not, oh, just wait a couple weeks, it'll pop up eventually. And so I found one trial so far in the neoantigen space that even barely starts to look like this. So there's, I think that this is like one patient who was like an exemplary response to this very gonzo vaccine that Gritstone Bio has been making, where they do a prime with an engineered chimpadenovirus, and then they have to boost with a self-amplifying RNA because you start getting anti-adenovirus antibodies, so you can't boost with that. It seems logistically really hard. But they did manage to get some like non-trivial circulating T cell responses against what they vaccinated against. So they got to like, 1/3% of circulating CD8s. So this is pretty good. Like this is what a successful vaccine would look like. My sense is that most of their patients didn't look like this because they didn't put them in the paper, but they at least had like an existence proof of our vaccine could sometimes be good. But the stuff that mostly goes into neoantigen vaccines, there's like a few regulatory glide paths. So poly-ICLC is one of them. Another one is montanide. These are like adjuvants that have been around for a long time. They have a really well understood safety profile. The FDA is just fine with using them for whatever. They just happen to not work. So people put them into their protocols because they know that the protocol will get like you can submit the IND and get an approval back pretty quickly because your reviewer is familiar with poly-ICLC and montanide, but none of those trials ever work. So there's kind of like a regulatory friction where you need to actually try new potentially dangerous adjuvants or vaccine formulations to get stuff that actually works. Like just basic vaccinology is unfinished here, which is weird because like vaccine design is a really old field. We just usually don't optimize for T cells. Most of what vaccinology has been is antibodies and clearing infectious diseases. So this just kind of like, we just didn't notice that we're bad at this particular kind of response, but we actually are, and the things that people think of as T cell vaccines aren't good for that job. So we have like a mouse study, it's this program we call Oyster, which I already forgot what it stands for. We do a lot of mouse vaccines and try to compare like bake-off adjuvants. This is like one particular round of Oyster where we have CpG, so this is like little nucleic acid adjuvant, and we have a sting agonist called DMXAA, and we have poly-IC, which is the core component of that thing the FDA really likes, poly-ICLC. There's a couple different model antigens here. Oh, this is wrong direction. So, okay, here are the T cell responses to these things. The green is a control, so it should be zero-ish. And then poly-IC is the yellow, which is sometimes a little different from the control, but it's not that different. And then CpG and DMXA both like kind of work. They're at least better than poly-IC. I don't know if they're like sufficiently better to be a useful adjuvant in people, but we've tried a couple other ones. Pretty much everything we try is better than poly-IC across big dose ranges and lots of different antigens. So there's like a strong default in my field, which just like clearly is suboptimal. So now we need to do more of this to sort between, we now have CpG, 2 different sting agonists, and mRNA all look pretty good. So we're applying to get funding to do more rounds of this to sort of rank those amongst each other to know what to put into a trial. But we know that it's definitely not the thing that people typically use. Like if you got everything else right but used poly-ICLC, the whole thing would probably still fail because the vaccine would just fizzle. You wouldn't get much of a T cell response. Okay, so the summary is you need vaccines that get really strong T cell responses. If you could get 10% responding to whatever you vaccinate with, that would be awesome. You need targets that don't look like self. Poly long read sequencing gives some of that to you. And then you also need to predict what's actually on the surface of a cell better than we currently do, which is probably being a lot more careful about looking at mass spec data and making immune machine learning models. And if you put all that together, it probably still wouldn't work, but I think this is the like